So now we're going to talk about gene regulation in eukaryotes. As I mentioned before, eukaryotes are going to be a little bit more complex than the prokaryotes, and the reason for this is because, first of all, they have a lot more DNA. Okay, prokaryotes are usually fairly small in their genomes, and typically the eukaryotes are going to be quite a bit more. Also, the DNA that's there in the eukaryotes is typically associated with proteins known as histones and other proteins as well. So in eukaryotes, the mRNA has to be able to leave the nucleus. Before it does so, there's a lot of things that happen to it. That mRNA can get spliced, there's a cap that gets put on the end, it gets a tail, a polyadenylated tail put on the end, and this all has to happen before it can leave the nucleus. The movement of those RNAs from the nucleus into the cytoplasm after transcription is very highly regulated, and there's a lot of things that have to happen to it before this happens, before it can leave. The mRNA stability also plays a role in how genes are going to be regulated, and then how the mRNA is being translated, the stability of the mRNA and other things like that, as well as how proteins themselves can be modified or processed, tags can be added to them, and they can be turned over or degraded. So pretty much at every step of the pathway, starting with the DNA and going all the way through to the protein products, this process can be regulated in eukaryotes. So we'll talk about chromatin remodeling. The DNA, remember, is going to be bound and associated with the histones. So that it's going to be associated here, and this will determine whether or not it can be translated, whether or not it's going to be accessible to the RNA polymerase. If it is accessible, transcription can happen. Transcription itself can be regulated. Whether or not the particular transcription factors are there to recruit the RNA polymerase to the promoter region, and so on. Once transcription happens, we'll have a pre-mRNA. This is the primary transcript. This needs to be processed in 3 different ways before it can leave the nucleus, before it's fully matured. There's going to be a cap that's going to be put on. There's a tail that's put on. It needs to be spliced and processed before it can leave through these nuclear pores. Once it has left, now it's in the cytoplasm, it can now be translated at the ribosomes. So the stability of this mRNA, whether or not it's being degraded, whether it loses that cap and that tail, whether or not the ribosomes are there to find it, and so on. This is all at the level of translation regulation. Finally, once the protein products are made, these proteins can be modified in many different ways before they can be fully active.